All right, guys. Last time we talked about some of the general properties of the non-covalent forces and non-covalent properties of aqueous solutions that are important for understanding molecular structure, right? Right. And I've, keep pointing out how important understanding molecular structure is from the point of view of understanding function in biology, right? That's why we care about it so much. Okay, and so like I said, the last lecture, we talked about some of the non-covalent um, weak interactions and hydrophobic effect that are important for specifying molecular structure. Today, we're going to start thinking about covalent structure, right? And how covalent structure and some of the properties of that covalent structure lead to other aspects of molecular structure that are really critical for the function of an important biological molecule. And the important biological molecule we're going to be worrying about today is, of course, proteins, right? So the question worth asking is, why study proteins, right? What's the big deal about proteins? Why worry about them so much? Why focus on them so much? Why are they so important? Right. So I'm going to answer that question a couple of different ways. One, of course, is that they're essential components, as many of you, all of you hopefully know. Proteins are essential components of basically everything that's alive, right? So life as we define it nowadays on this planet doesn't happen without proteins, right? Okay, that's one reason, but we can probably be a little more specific than that. So the reason they're so important is because they perform so many of the critical roles, the critical functional roles, there's that word again, we associate with life. For example, right? Um, just fundamentally, right, is we're structure building upon structure. Organismal and cellular structure depends on proteins. You can't build all of the parts of a cell and the things that stick cells together in tissues and all of that stuff, everything we think of. It's not only protein, but without protein, it doesn't exist, right? So they're critically important for that. Remember all that information storage stuff I alluded to that we're not going to study, unfortunately, because we don't have time. But all the stuff you've been exposed to before in information storage and retrieval, right? The packing of all the information into a organism's genome, the reading of that information back out again, all the regulation that goes into deciding when that information goes in and comes back out. All of that depends upon proteins. Without proteins, it doesn't work, right? None of that works. Even though the storage medium is nucleic acid, without proteins, you got no storage and retrieval systems in the biosphere on this planet today. Um, Third reason, right? So material recognition. Right? And what you could also call recognition and transmission. Just finding stuff and moving it around. Doesn't work without most of that. The vast majority, not all of it, the vast majority of it happens because of proteins or there's a protein involved. Some other biomolecules, nucleic acids, and carbohydrates do some of this too, but the lion's share of this is done by proteins, right? A biggie, a really super biggie, catalysis, the ability to drive chemical reactions much faster than they would spontaneously go. Depends at the, 90 something percent level, 95, 98% level. Depends on proteins. And even in situations where nucleic acid, as you guys may know, is involved in doing catalysis, in most cases, there are associated proteins. If you take the proteins away, that nucleic acid catalyst doesn't work anymore, right? So they're critical for catalysis. And then finally, what I call energy transduction. And in this case, this is a matter of where we take something like chemical energy, something that's stored as a chemical, and we turn it into mechanical energy to do work, right? For example, that little listeria cell motoring about in the cell is taking chemical energy and converting it into mechanical energy so that it can motor about the cell and go find other cells to infect. All right. Now, these last three guys involve a cascade of functions that I'm going to use sort of over and over again. We're going to start at the basement and we're going to build our way up, right? These three in particular sort of build in a cascade from what I almost call binding, right? Material recognition. And we're going to build on binding to then do catalysis, which proteins is, of course, related to molecules you guys know called enzymes, hopefully, right? And putting both of those guys together, binding and enzymes in fancy ways, leads to energy transductions. And a good example of this, of course, are things like molecular motors. And we're going to see one at the very end, right? Okay, so hopefully that's a pretty good reason to motivate some of the reasons why you care so much about proteins, why we dwell on them so much. But as another way to illustrate this, consider a quick example of the power that proteins have as agents that can do this cascade of function. It's so important in biochemistry. All right, here, I'll give you an example. Remember that I mentioned in my previous lecture, or I've been using it a couple times, you're going to see it many more times, this that initial reaction at the top of intermediary metabolism where we make glucose 6-phosphate, right? And I pointed out that that reaction, being non-spontaneous but essential, is driven by this reaction, right? Taking ATP and hydrolyzing it to ADP and inorganic phosphate, right? And we couple this to glucose and inorganic phosphate is how we get glucose 6-phosphate, right? But if you think for a second about this reaction, right, the hydrolysis of ATP, and how important that is, turns out that much of the energy stores in the cell, not all, depending how you define it, but the large lion's share of all the energy stores that cells rely on to do the stuff they do, is stored in one of two nucleotide forms, all right? Either ATP or ADP, excuse me. Caught that error, or GTP, right? Okay, well, think about this for a second. If you consider the structures of ATP and GTP, and they're shown right there, we know if we write down our little, classical organic chemistry Fisher diagrams with bonds indicated by lines and whatnot, we can recognize the difference. We can see quite readily the presence of, sort of this exocyclic amine group here and here is different, and there's this aromatic carboxyl group, and so they look different. And so I show you there the CPK, sort of the structure of these molecules would look like in their van der Waals radius, and put those into sort of a rotation for a second here. And you look at those and you're like, what if I put you in a dark room and I 3D printed those two molecules and handed them to you? And it was completely dark. And I asked you to tell me which one's ATP and which one's GTP. How long do you think it'd take you to figure it out? Knowing what I know about these, I might have, It'd take me a minute or two, maybe a little longer. I don't know. I'd eventually feel my way around. If I knew that was the goal, I'd probably eventually figure it out. Maybe 2, 3, 4 minutes, something like that. Here's the amazing thing. The proteins, the enzymes that do that reactions like the one I've been talking about, they can discriminate between GTP and ATP with something like near perfection, right? They will actually work on ATP and they will not work on GTP at all, right? Or, you know, parts per billion will they actually slip up and use GTP. So the fidelity is phenomenal. And they can do that. They can discriminate between the two of those things in a fraction of a second, right? Sub-millisecond timescales. They can tell the difference, right? So that's just remarkable, right? And so a fascinating thing, and one of the things, you know, sort of we really want to try and understand is how is that kind of molecular recognition possible? And it has everything to do with the nature of proteins, how they're constructed, their three-dimensional structures, and that all starts at the properties of the underlying constituents, which we of course know are the amino acids.